Okay, so today we are learning about DNA replication. So first we're going to start with DNA structure, just because if you know a little bit about the structure of DNA, it really helps to understand the way that it replicates. So DNA forms a double helix strand. So there are two strands of DNA that kind of coil around each other, and I'm sure most are familiar with that shape, but there are kind of three parts to the one DNA strand. So first we have these white hexagons are representative of the nitrogenous bases, and then each of those has a pentose sugar attached to it. Pentose sugar, which would be a 5-carbon sugar. And then each of the 5-carbon sugars have a phosphate group attached to them. So those are kind of the three parts. And these nitrogenous bases, so these are the adenine, thymine, guanine, and cytosine that kind of pair up together. So adenine always pairs with thymine, guanine always pairs with cytosine. And that's because they are shaped in a certain way that they can connect at these corners with each other. So G and T could never pair and C and A could never pair. So they make a hydrogen bond together, which is kind of like a weak attraction between molecules of different polarity, a positive and a negative kind of a thing. But it forms an attraction that is somewhat weak if it's just one, but when it's all of them, it makes a pretty strong bond. So again, this kind of curves into a double helix structure where you have the nitrogenous bases are kind of like the stepping stones of the ladder, or the bars of the ladder, and then it would have a sugar phosphate backbone. So next we can talk about the replication of DNA. So DNA replicates semi-conservatively, which means that one of the strands is an original strand, and then one of them is a new strand in each replicated DNA. So here we have a leading strand would be the one on top, and on the bottom we would have the lagging strand. So the leading strand runs 5' to 3', and the lagging strand runs opposite that, which we got these numbers wrong, those will be fixed later, 3' to 5'. So those are kind of mirror each other. So first step in DNA replication, you have a bunch of different enzymes that kind of come in and do their part. So the first enzyme is helicase. So helicase kind of unzips the DNA, so it breaks the hydrogen bonds that are keeping these two strands attracted to each other, and kind of unzips the double helix strand. So second, we have SSBPs, which are single-stranded DNA binding proteins. So those kind of bind to the DNA and they keep them separate. They keep them in place so that they don't at the right over here, after helicase goes through, they don't reattach to each other. So first we start on the leading strand, and DNA always, the new strand is built from its 5' end to its 3' end. And again, it mirrors the leading strand. So it starts on the leading strand's 3' end. So first DNA primase kind of sets a starting point and says, okay, we're going to start here. And then DNA polymerase will kind of fill in based on the opposite nitrogenous base, the opposite nucleotide that it's pairing up with. So it will have A's go to T's, G's go to C's, and it kind of rebuilds the strand backwards. So then, once it's done, this 5' end on the right would be the same as this 5' end on the left of the leading strand. So then the lagging strand, again, this should be 3' on the left, 5' on the right, fills in just as the leading strand, except it has to go in chunks because this helicase is slowly unzipping the molecule. So it will, the primase will kind of be like, okay, this is what we have unzipped so far, so let's fill in this area with DNA polymerase. And then the helicase will unzip a little more, so the primase will go back a few and say, okay, now let's start here. And so it kind of builds the new strand in these fragments called Okazaki fragments. And again, the reason it has to do it this way is because the lagging strand starts on 3', and DNA can only fill 5' to 3'. So this purple one, the 5' end is on the left here. So it has to kind of fill in chunks because it's filling as the helicase is unzipping. So again, these are called Okazaki fragments, and they only happen on the lagging strand. So then one of the last enzymes that comes into play is the DNA ligase, and this one kind of zips the strands back together, these little fragments, into one full strand on the bottom here. So DNA ligase is the one that does that. So these are the steps that we have. We have helicase unzips, SSBPs kind of hold the strands in place. DNA primase sets the starting point, and DNA polymerase fills in the gaps, and DNA ligase connects the fragments together. And then you have 2 strands, and if everything goes according to plan and there aren't any mutations or any errors, DNA polymerase also proofreads as it goes. So if there aren't any errors, then you would essentially have 2 strands that are identical to the first original strand. One last one, we have telomerase, which creates telomeres. These are ones that you're kind of born with your DNA, and as your DNA is replicating when you are being created, from an embryo to an actual human, as your DNA is replicating in these stages, it will put telomeres on the edges of your DNA, on the ends, almost like a shoelace, plastic shoelace cap, to keep it from unraveling. So telomeres are actually related to age. So there are certain things like smoking, obesity, poor lifestyles that can actually shorten your telomeres faster than they would be if you were just living a healthy life. And an increase in telomeres is also associated with an increase in cancer sometimes. So sometimes they will artificially add on telomeres to cells that should be dying. So anyway, that's just a side note, but telomeres there, cap it off and make sure your DNA lives as long as it can.